All right, we're going to get started. And just a quick note to all of you joining up today that we're going to have a slight change in the link that you use to join up on the meeting. And we're going to post this to our website, which is dfhcc.harvard.edu/connectscience. We're also going to send out an email notification over the weekend with the new link. If you have not yet signed up as to get notifications, please go to that URL and put your name in to receive those email notifications. Thank you all very much.
Okay, so this is Nelly Poyak from Dana Farber, and thank you everyone for joining us at this seminar, and also thank you for the FHCC and the Leukemia Lymphoma Society for hosting these webinars. And as Richard already mentioned, the website that you see on the first slide here will have the talks also uploaded, so you can look at the past presentations as well. It usually happens at the end of the day. And then just as a reminder, there is a Q&A on the Zoom that you can submit questions, and we will answer as many as we can during the live presentation at the end of the talk, and the speaker will have access to these questions and he can get back to you to answer them later on. So it's my pleasure to introduce today's speaker, Ross Levine from Memorial Sloan Kettering Cancer Center. Ross is a Lawrence Joseph Dinan Chair in Leukemia Research and also the Director of the Center for Hematologic Malignancies and Chief of the Molecular Cancer Medicine Service at Sloan Kettering. And the title of his talk is Deciphering and Targeting Clonal Evolution in Myeloid Malignancies. Ross, thanks for joining us.
Great. Thanks, Nelly. It's really an honor, and I really appreciate what Nelly and Dave and Richard and DFCI and LS leadership have done. It's really great to be able to connect with our community and our colleagues, and it's a privilege to present some of the work that we've been doing recently to all of you today. You can see in the bottom left my Twitter handle, and I would encourage all of you, if you have questions after the talk, to just shout out on Twitter, and I promise you that we'll get back to you. Here's my list of disclosures. None of them are relevant to any of the work I'll talk about today, except for the fact that I'm on the QIAGEN Supervisory Board of Directors, and obviously we do extract DNA to do the studies I'll talk about today. Before talking about science, I thought I'd say a few things about the world we live in right now. I think it's really important for young investigators and trainees to understand that we realize how challenging this is, and we're all going to do everything we can to support you. And there's a lot to say and think about people that are applying for fellowships and looking for jobs and trying to get their careers started. And we, as a community, figure out ways to support everybody in that endeavor. To our colleagues who are doing clinical work, many of us will take our time on the clinical side. We say thank you, and to all the healthcare workers, and everybody stay safe. And then just a couple of comments. The first is that no one's being productive. It's really hard to do work at home. Young kids, without kids, the distractions, CNN on time, all the time. No one planned for this, and just do the best you can. Stay connected as a lab with your community. You know, that's much more important than that grant or that paper, and try to focus on small goals. And last and most importantly, find ways to contribute. Review grants and papers, talk to a new collaborator, do a meeting with a mentee or mentor outside your local environment. I think if we all find ways to pitch in, we can make a difference. So before I show data, I thought I'd start with the acknowledgement slide. All the work I'll talk about today was done by this amazing group of people in my lab, largely done by Bobby and Lindy, who really are incredible visionary scientists, and it's a privilege to work with them. And they've had a lot of help from a lot of people in the lab, including Tiffany and Nicole. Mike Wartz has done some of the work on CRISPR I'll talk about, and Aaron Vinny was involved in a lot of this work. He's on the job market. And we've got lots of great collaborators, including Jason Butler at Hackensack Starmeyer, Martin Carroll in Philly, Giannis at NYU, Todd Drulli at WashU and Archer, and our colleagues at Mission Bio who've been incredibly helpful. So my laboratory has been studying leukemias for the past 12 and a half years since I left my training at Dana Farber. And the reason we've decided to focus on AML is that it's a disease where, although we now understand the biology substantively, outcomes have not really improved really dramatically. And on the left, this is data from MD Anderson making the point that despite there being a p-value that outcomes have improved, it doesn't really meet the sort of, have they improved in a meaningful way? And on the right is data from Peter Valk and Ruud Delwell, where they did gene expression analysis of the HOVON AML cohort. And the reason I put this up is that not only is AML a disease where outcomes are bad, it's also probably 15 to 20 distinct diseases. And this is gene expression profiling that makes the point that there are probably 15 to 20 AML subtypes. And so the challenge is not how do we treat AML in block, but how do we separate AML into different subtypes and direct the right therapies to those specific subtypes based on mechanistic understanding. I'll also point out that AML is a disease of the aging population. The median age is 68, and AML is a hard disease to treat in younger people, let alone in older folks. And that's because both the disease is more aggressive and less responsive, and comorbidities and age make it challenging. And so one of the questions I think in AML is, why is AML so hard to treat, and why is it so biologically complicated? And there's been lots of data to suggest that it's been a tough nut to crack. But on the other hand, from a genetic perspective, AML is a pretty simple disease. This is the data from TCGA, which makes a couple of really important points. The first is that there are only 50 to 70 recurrently mutated genes at more than 0.5%. So the world has developed reagents to study basically every recurrently mutated AML gene. The second is there are only 3 to 8 coding mutations per AML genome, at least within the coding genome at NGS detectable levels. And what that means is that there's tremendous complexity in the disease with genetic simplicity. And so the idea that we really have sort of been obsessed with for the last few years is how does a small number of mutations in different combinations of permutations lead to very different outcomes in different patients? And the analogy we like to use, and some of you may appreciate, as I was watching last night, Bridesmaids with my daughter, is the Judd Apatow movie. Every Judd Apatow movie has the same cast with slightly different permutations of which actors and actresses, and a slightly different plot, and you end up in a different story each time. And think of AML as like that. You have 50 to 70 potential players. We mix and match them in every patient in a different way, and you get a very different leukemia. The other thing that we've learned in the last few years is that AML is an evolutionary trajectory that goes from normal stem cells to a pre-malignant state we now know as clonal hematopoiesis. Some people like to call it ARCH or CHIP. You can use whatever terminology you prefer, but just makes the point that we know that a significant fraction of acute leukemia arises from a pre-malignant state, which probably goes on for years with these early mutations in genes like DNMT3A or TET or ASXL1, enhancing the fitness of stem cells, but not causing transformation. Now, these obviously cells are not benign, and we know from Ben Ebert's work and a number of other labs that this predisposes to cardiovascular disease. But from my standpoint, what this means is that we have a genetically identifiable, through non-invasive sequencing of the peripheral blood, a risk factor for leukemia years before people develop it. And obviously, folks at the Farber, folks here at Sloan Kettering, lots of places have set up clinics to begin identifying people that don't have malignancy but are at risk. And we need to move from identifying risk patients to ultimately treating them. And I'll talk about that towards the end. The idea that we have to not only identify people at risk for heme malignancies, but ultimately intervene. But there's a more fundamental question which I want to spend the first half of my talk on, and that is what I call the math problem. There are probably 20 million people in the United States who have pre-malignant hematopoiesis that have these early mutations. Half of them have mutations in one gene, DNMT3A, but there are fortunately only 30,000 patients per year that develop an overt myeloid malignancy. So what does that mean? That means that this pre-malignant state is not rate limiting, that it's occurring at a much higher rate than transformation. And that means we need to understand what are the events that occur after those early events that enhance the fitness of stem cells. Not only what are the events that drive this early clonal expansion and the clinical outcome and this idea of intervening, but why are there only a small fraction of people that develop leukemia? Is it the order of events that has to occur? Is it the specific stem or progenitor cell subtype that acquires mutations? Is it the microenvironment, the microbiome, immunological surveillance? And then last and not least, we need to understand, what do we target? Do you target the early events that induce or initiate the process, or should we be only targeting the things that cause transformation? I'll point out that Scott Armstrong had a paper in Science recently targeting the idea of NPM1 as not an initiating mutation, but a transforming mutation. And we need to figure this out more broadly. And so we think of AML as like a Jenga puzzle. What we need to do is use better and better technologies to delineate the genetic and epigenetic events and cell factors that give rise to AML, but then understand which are the ones that we can pull out of the leukemia to really get to dependencies. And I'll try to get to this concept at least in part. And all the data I'll show you today is unpublished, and we're thrilled to discuss it with all of you. And a lot of it is in a recent preprint. And I'll encourage all of you, if you have questions after this talk, to ping me, Lindy, or Bobby. And so one of the questions in AML is, can we put together a sort of map of all of the events that go from a normal stem cell to fully transformed cell? And the way people historically have done this is through bulk sequencing using variant allele frequency to predict the order of mutations. This is data from Peter Campbell and Ellie Papamanuel and colleagues showing that using variant allele frequency, we can figure out what are the early events, the sort of mid events, and the late events in leukemia. The problem, though, with this data is that it's not granular on a per clone basis. And it really doesn't give you, with tremendous resolution, the specific genetic events and the order and what's happening in different clones. And so from our standpoint, there's a lot of things we need to better understand about this process of clonal evolution. Can we better delineate the sequential events that occur from normal stem cells to pre-malignant stem cells to overt myeloid malignancies? Can we understand what are the key events that drive that? What are the things that not only occur, but induce clonal expansion? Can we map the trajectories from early to transforming event? How does the milieu of clones change during transformation? I'll point out that Nellie's work in breast cancer has really shown us that many cancers are an ecosystem of different clones. And we would argue that in leukemia, we need to delineate that clonal architecture and then go back to the lab and do the similar studies in hematopoietic malignancies. And then last, can we target this? And so we believe the answer is that you need single cell analysis of clonal architecture and single cell profiling. It's just not the same single cell RNA sequencing that you read about each week in Nature, which is really important. But we believe that understanding clonal analysis at single cell level is critical. And so about a year ago, we set out with our colleagues at Mission Bio to develop a custom amplicon-based methodology using their Tapestry platform, which allows us to look at this set of genes in about 150 patients with AML. All of the data is in this preprint. You can see right here, and I'm happy to share the link after. And we also married, as you'll see, with the immunophenotypic profiling, so you can look at mutations versus cell state. And Koichi Takahashi and colleagues from MD Anderson have really nice complementary data in a preprint on bioRxiv, and I'd encourage you to reach out and check out Koichi's work as well, which is really seminal. And so what did we basically do was enumerate in 1 to 10,000 cells per patient the mutations in every single cell. And what you get when you get this data is a map like this, where you get clonotypes, with this as being the cell number and this being the genotype. And you get anywhere from this clone, which has these 2 mutations and a small number, to this large clone, which is the dominant clone, which in this case has an IDH2, STAG2, and NRAS mutation. And in about 146 of our 150 samples, we had enough cells where we could do clonal mapping. And I'll make the point that we don't think we're at the asymptote for this technology. We think 1 to 5,000 cells isn't enough, and I'll come back to that. But at least we can begin to look at what the data looks like. And so what did we do? We did a subset of different samples. We did a small number of clonal hematopoiesis samples, largely focusing on samples that had more than 1 mutation, because there was no reason to map clonal diversity if there's only 1 recurrent mutation. We looked at myeloproliferative disorders. We looked at leukemias that had signaling mutations without epigenetic regulators, leukemias that had DNMT3A, TET, ASXL1, IDH mutations, and then layering on FLT3 and RAS mutations. You can see the number of mutations goes up modestly, but not dramatically. The number of clones goes up, but ultimately, what we observed was not tremendous changes in genomic complexity if you were to look at bulk numbers of mutations or even numbers of clones. We then looked at clonal trajectories in different disease subtypes. So this is what clonal hematopoiesis looks like. When we see multiple mutations, we see that generally they're in different clones. And so this is an example of a clonal hematopoiesis subject that has 3 different mutations, and they're actually all in different clones. So when you see a CH patient that has 2 different DNMT3A mutations, by and large, the rule is that they're in distinct subclones, and that's an important point. In contrast, in AML, what we saw was 2 things that were different. The first was accumulation of mutations. You'd expect that as you go to overtransformation. But the thing I think that we didn't expect was that the majority of AML patients, 86% either had 1 or 2 clones that we call a dominant clone, and we define the dominant clone as being more than 30% of the cells. Why is this important? That means that, for example, in this patient that had these 3 mutations, we don't see any cells that have 1 mutation. What that means is the initiating clone that has whatever the first mutation, maybe the DNMT3A mutation, is not seen. And that's one of the reasons why we think 1 in 10,000 cells is not sufficient resolution. As these technologies get better, we can go deeper and higher throughput, we'll get better data. But what it also suggests is that specific combinations of mutations may lead to clonal advantage over all of the antecedent or parallel clones, either because they're fitter, they proliferate more or die less, or maybe they secrete factors that suppress the dominance of other clones. I think the latter may in fact be the case, and it's an important point because if you look at AML, and there's really nice data from Laurent Lish and John Dick on this, that for example, antecedent clones are easily detectable a month after chemotherapy. So if these clones are easily seen as soon as you clear the leukemic bulk, that means it's not that they're not there, but that they're suppressed. And we think this is an important biological question. We then looked at clonal diversity using a Shannon diversity index, and what we can show is with disease progression to a more advanced disease, we get greater clonal diversity. And in fact, although there are dominant clones, the size of the dominant clone is smaller. And what that means is as you go to a more advanced disease state, you get a clonal dominance, but it's a less dramatic clonal dominance, and you get more clones going on. And that means we think that the different disease subtypes and genotypes and settings may have different clonal architecture and milieu, and studying different disease states may be critical. We then wanted to know, are different mutations in the dominant clone or not? And this is not whether they're the first mutations, whether they're in the dominant clone. You can see that, for example, CALR mutations or JAK2 mutations in NPNs are virtually always in the dominant clone, not surprising. And for example, NPM1 is virtually always in the dominant clone. Other mutations like KRAS or FLT3 are almost never in the dominant clone. And then you get interesting things like TET2, where half the time it's in the dominant clone and half the time it's not. And that suggests, again, that some mutations may always be driving the disease and always be in that large dominant clone. Others may be late comers to the party and may in fact be minor clones. And others may have a pleiotropic role where sometimes they're driving dominant clone expansion, sometimes not. But this is looking at individual genes. What I would argue here is that this is not the best way to think about this. We need to look at combinations. And this, begins to look at combinations. On the left, this is looking in the bulk data at co-occurrences or exclusivity. On the right, this is looking at a per clone basis. And again, in 150 samples, we don't think that you can actually look at inclusion or exclusion, but as we all generate bigger and bigger maps, we'll be able to get patterns that tell us things. But we began to look at cooperativity and exclusivity and said, are there any sort of rules that we see in our data? And the first thing that we observed was that epigenetic mutations in the big 4, DNMT3A, TET, IDH, and ASXL1, when they co-occur, they virtually always are in the same clone and they're in the dominant clone. So that means is you don't want to study DNMT or TET or IDH or ASX1 by itself. You got to study how these things work together and understand on a global and locus specific level how the dysregulated gene regulatory patterns and promoting clonal dominance. And by contrast, if you look at signaling mutations, as you'd predict, these are generally in distinct clones, suggesting that in fact signaling mutations are driving parallel clonal evolution. There is an exception, for example, for JAK2 and NRAS, where NPN clones that have JAK2 that then require NRAS, the NRAS mutations are occurring in the JAK2 mutant cells, suggesting those cells tolerate and actually maybe favor having those 2 mutations together, which we would not have predicted from bulk sequencing data. And you can even show that different combinations of mutations have clone-specific co-occurrences. So this is drilling down on DNMT3A and IDH mutations and the co-occurrence. And what you can show is that JAK2 mutations only occur in IDH mutant clones, whereas the JAK and RAS mutations occur in the double DNMT IDH double mutant clones, and the FLT3 mutations only occur in the DNMT3A IDH mutant clones or in the IDH1 clones. And what this means, if you're going back to the lab to study what these genes do, you need to engineer the right clonotypes or combinations to study the biology, and not what is this mutation or that mutation or even these pair do alone. We then wanted to really see if we could walk through the history of these leukemias from normal stem cells at the top in gray to the early events to the late. And so what we did was develop a Markov decision model to map the likely trajectory to AML. And you can see this is the predicted order of events based on the genomic data. And then the size of the circle is the size of the clone. This lets you do two things. You can see the likely trajectory to leukemia, and not only the trajectory, but when, boom, does the clone expand and actually give you clonal expansion or dominance. And so we asked a couple of interesting questions once you have that data. The first was, do some mutations, do they have a role in initiation versus clonal dominance? And you can see again that JAK, IDH, DNMT3A are initiating mutations. By contrast, RAS and FLT3 are late mutations. But you get two other flavors of mutations that are interesting. One is TET2, which is pleiotropic. It can be initiating or it can be late, which is kind of interesting. So you'd want to ask, what does TET2 do in different trajectories at different times? And then you have NPM1 and to some degree ASXL1, where they're like the second person to the party. They're never the initiating mutation, but they're always the second or third mutation, probably because they can't start the process. But once they do, you get clonal dominant. And that suggests a very strict to these mutations. And then we wanted to go back to genomic data. We predicted the initiating mutation. Could we see single mutant clones that are genetic proof of initiation? You can see that in blue, we do see it for a subset of cases, and it matches up to our predictions quite well. But in gray, a lot of the cases we don't single mutant clones. This means we're just not getting enough sequencing data. And we'd argue in the next couple of years with better and better single cell DNA sequencing data, we'll be able to see that data. And that's really important. By contrast, we also see some interesting things, like, for example, in DNMT3A mutant clones, the non-RNA2 clones actually are likely to be initiating, but the RNA2 clones are not, either suggesting that RNA2 don't start disease, or more possibly, they actually, once they occur, they cause rapid transformation to leukemia. We then asked if specific combinations of mutations induce transformation, and whether they cause clonal dominance. And you can see here that, for example, DNMT3A and IDH mutations, when they co-occur, they dramatically cause clonal expansion, the same for NPM1, FLT3, but NPM1 and RAS don't actually give a fitness advantage. In fact, in some cases, it's worse than either mutation alone. And this, we think, is the next phase of cooperativity to understand mechanistically what combinations not only co-occur, but what are the combinations that cause clonal fitness advantage versus others, and why? And we need to go back to the lab, as I'll talk about, to understand that. We also can look at clonal evolution during therapy, so we've begun to do that. These are a couple of patients who had FLT3 mutant disease, where we can look before and after FLT3 inhibitor therapy. You can show that in this patient, before therapy, they had a STAG2 FLT3 clone as one of the dominant clones. But as they relapsed, you can see that you get expansion of other clones, like in this case, a RAS clone expands that was pre-existing. You get these RAS and RUNX1 clones that weren't there before. But this patient's a great example where you basically show a clonal switch, where during or pre-gilteritinib therapy, it's these clones, and then you get this very clear clonal switch clones, virtually all of them new RAS clones that were not detectable single cell before. So you can begin to use this as a way to map the emergence of many resistant clones. And it's not going from one clone to another, but it's a clonal sweep as you get clinical resistance. The challenge, though, with all this data is that it's all genotype. And you want to be able to link genotype to phenotype. And you can do this in many ways. And we think one way to do this is to link DNA to protein and to look at immunophenotype versus genotype. We also are beginning with our colleagues at Mission Bio to pioneer genotype to transcriptomal alterations, and we'll have a lot to say on that when we all get back from this unfortunate hiatus. And so what we do, very similar to CiteSeq, is marry the genotype to bar-coded surface molecule mapping, so you can look for every single genotype what the cell surface immunophenotype. And this is a leukemia patient. You can see you have one set of wild type cells, and those are CD3 positive, and those are the T cells. So in this patient, without having even a matched normal, you can identify the wild type cells and compare them within an admixed bone marrow sample. You can do lots of really interesting things. So the one thing we've done is take patients, in this case, a patient that had DNMT3A mutation, you could show that this 3A mutation is in the myeloid cells and in the T cells, but not in the B cells. So for this CH mutation, it drives two lineages at the expense of another. This is another 3A mutant clonal patient that had two mutations. And for one of the mutations, the R635Q, it's in T cells, and it's in a small number of myeloid cells, but it's not in B cells. But the RA2 is in the myeloid cells and the B cells. You can see we can basically map all of these pre-leukemic mutations within the sort of ontogeny of hematopoiesis. And I think this will be really fun to do moving forward. We then went back to leukemia and asked, can we map mutations on complex immunophenotypes? And what Bobby and Lindy did was to develop a neighborhood analysis to basically map all of the different mutations within different immunophenotypic subclusters. And in this patient, what you can observe is they had a bunch of different clones, including FLT3 and RAS mutant clones. But the RAS mutant clones, in this case, you can see drive a very specific immunophenotype, and that is associated with CD11B expression. So in this patient, you have different immunophenotypic clones, and the RAS clones have a very specific immunophenotype different from the FLT3 clones or from the 3A only cells. And this suggests potentially, for example, if you had a cell surface targeting molecule, you could target these specific clones. And we think that the idea of marrying this to immuno-oncology will be fun to do. We hope to collaborate with a lot of you moving forward on that. We also can show that if you look in different patients at this, that the dominant clone often in 8 of 14 patients has a different immunophenotype than the subclones, suggesting that you might be targeting different cell surface molecules in the bulk leukemia versus these different subclones. You're going to need to do this at significant resolution. And the last thing we showed was that different mutations, in this case, for example, RAS mutations alone or in combination with IDH, promote the expression of CD11B, whereas IDH GNMT3A mutant cells are CD34 positive. And so you can begin to map different mutations and combinations on the immune surface repertoire. And there are lots to do on this moving forward. You can add RNA. You can do hematopoietic markers, including, say, the Ravi Majeti, Irv Weissman sort of landscape of stem progenitor markers. And we're excited about the idea of bringing this back to colonial hematopoiesis and solid tumors. We work with Elizabeth Coleman and Larry Norton, and we can take admixed tumors that have hematopoietic cells and epithelial cells, and without any sorting, basically enumerate mutations in different subtypes. So lots to do on this moving forward, and we'll lots to chat about with lots of you. And so in summary, from this first part, single-cell DNA sequencing really allows us to look at clonal architecture and disease evolution. We can look at this with disease progression, therapeutic response, and we can begin to link genotype to phenotype. There's so much more to do with this technology, and there's lots of other groups doing great work in this space. Single-cell data to clinical outcome, looking at MRD, serial samples, RNA, and I'd encourage all of you to really get in on using technologies like this moving forward. But what we're very excited about is to go back to the lab and take this data, which lets us look at order and clonotypes and progression and model it in the lab. And so one thing Lindy Miles has done, who really pioneered this work, was begin to map order and ask, does order matter in the lab? And so from all of our data and other data, we know that TET2 and NPM1, that generally TET2 occurs before NPM1. And so she basically developed the first generation mouse model where she inactivated TET2 with CRISPR and then activated NPM1 using a CRELOX or FLT3 allele, the FLT3 allele from Jen Trowbridge. And what we're able to show is that if you do the order the way it occurs in patients, you actually get a fully penetrant leukemia, whereas if you do the wrong order, you get a much more inefficient leukemia. This gives early data to suggest that order does matter. And we think that going back to the lab to understand does order matter allows you to ask very interesting questions about epigenetic effects. Do specific mutations license for other mutations? Are there specific reasons or cell types or order matters? And lots to say on that. But ultimately, to do that, you need a new toolbox. And so what we've decided to do in the lab is to develop new tools where we can engineer mutations with different genetic control, with different cell types and ultimately using different inducible markers. And Bobby has really taken this to levels that are going to be really awesome. And I'll encourage all of you to see Bobby give talks in the next couple of years, and ultimately when he goes on the job market in a year or two, to really hire him immediately. Because he's going to really be pioneering this awesome work. And just a shout out to Bobby and Lindy, who are really the visionaries here. And so let me give you an example of some of the tools we're developing. So some of you may know who are in the leukemia field that all of the FLT3 alleles that have been developed by Don Small's lab, by Derek Gilliland's lab, when I was there, Ben Lee led this work, were germline FLT3 alleles. And that's great. And those are the alleles that we all have been using for the last 13 or 14 years. The problem is that FLT3 is the last event, and it's not a somatic FLT3, although Don's allele can do some of that. And so what we did was develop a FLT-inducible FLT3 allele, which allows us to activate FLT3 after other mutations. The allele works beautifully. You can see the recombination. It's quite tight. It works quite well. And we can show that by itself, you get a myeloproliferative disorder, which is nice. It's actually much more penetrant and rapid than with the germline, suggesting perhaps that when you somatically activate it, you actually can bypass some of the in utero feedback mechanisms that might limit the expansion of FLT3 mutant cells. But the most exciting thing is that by contrast, unlike with the germline FLT3 plus NPM1, which gives the disease, but it takes over a year to develop, look at the latency if you activate FLT3 and NPM1 either together or sequentially. So we think that that's an important point. The disease looks just like the human disease with CD34 negative, fully penetrant leukemia. And so we'll have a lot to say about this moving forward. We think that it'll be exciting to develop models that really accurately recapitulate the series of events, the clonal milieu, and ultimately, if we can use these systems to uncover mechanisms, we'll go back to the clinic with those insights. But the last part I want to end with for the last few minutes is, how do we understand this from a targeting perspective? The fundamental problem in AML has been a dearth of therapeutic targets, except for signaling effectors and IDH, and obviously really awesome work on venetoclax. We need to understand what these mutations do to target them better. And so we noted that there was a fundamental problem in the field, and that was that these early epigenetic mutations, we don't understand why they induce the advantage that they do to stem cells. And this is despite 10 years of mouse modeling from my lab and many others, lots of RNA-seq, describing that. We just don't understand how these mutations enhance stem cells. And every experiment we do is an in vivo experiment. We engineer a mutation, we have to wait a year and read it out. And that's just not amenable. And so we thought you need new systems where you can study these mutations at scale and perturb them. And so we tried a lot of systems, but the system we've really settled on is one developed by Jason Butler and Shaheen Rafi, where they cultured normal stem cells on AKT-expressing stromal cells, and were able to basically expand normal stem cells on the left, and on the right, passage MYELO leukemia cells. And so we're not sort of a normal hematopoiesis lab, at least it's not our sort of the fairway that we drive down in our lab. And so we thought this could be a system where we could take all the different mutant lines we develop and study them for 2 to 3 weeks. And importantly, in this system, you can propagate it in vivo after extended culture. And so the first thing we did was show that we could maintain stem cells from mice, just similar to what they reported. We used a FLT3 reporter to show you could maintain FLT3 negative, really long-term stem cells. We could get 500 to 100-fold expansion of stem cells. We get 100,000 or greater expansion of differentiated cells, and we can transplant these in primary and secondary recipients. So the system works really well, but this is all just showing that we can recapitulate what Jason and Shaheen did. The reason we're excited about this system is that we can basically take wild type and mutant stem cells for your favorite leukemia or CH gene, plate them on stroma, and then ask, what does it do? And then perturb the system. And so this is what happens when we culture TET2 and wild type cells in the same system. We can map where the TET2 cells accumulate versus wild type cells and the more primitive cells. We can inject them in vivo and show the TET2 cells are advantaged, and they get myeloid and B-celled at the expense of T-cell engraftment. We can do similar studies for the ASXL1 knockout mouse model, show that you get expansion of long-term stem cells, so it's a more proximal accumulation versus the TET2. And we can do single-cell sequencing and begin to identify transcriptional effects in different populations in these cultures and inject them in vivo and show the retaining biology. But what we're most excited about is to take these different alleles and to begin to do perturbation screens. And this is data from Bobby, where in about 3 months, he can take all approved AML drugs and even combinations and begin to screen for effects on survival, differentiation, or repopulation capacity in these screens. And we're doing these screens now, or we were until COVID. Fortunately, we have a lot of data to analyze while we're out, and we'll have lots to say on this. 2 other things you can do in this system. One is you can knock in and knock out your favorite genes. So you can do tamoxifen in vitro in this system and delete TET2 and get the same phenotypes in vivo. So this is a lot like, using organoids. I've had the opportunity to see Charles Sawyers and Yu Chen talk about prostate organoids for years, where they now can basically get in vitro phenotypes, which give them scale and throughput that, versus doing 1 million mouse experiments. This also is possible with this system. But I think the thing we're most excited about is the concept of functional genomic screens. And so Mike Wartz has now optimized doing CRISPR in this system. And you can either do this on the top with constitutive Cas9 mice, or on the bottom with transient Cas9 electroporation, where we don't even need to cross the Cas9 mice. And this is a CD45 CRISPR in his system. He can show that he gets very efficient, 60 to 80% editing in vitro, in stem cells, and in differentiated cells. And more importantly, he's begun to take libraries, and this is one from Chris Vopkash's lab using an epigenetic library. We can basically maintain representation up to 14 days in this system and then inject it in vivo. And we think this is going to be really powerful, not for right now genome-wide screens, but for targeted screens or for domain scanning. And again, we'll have a lot to say on this moving forward. So with that, in conclusion, mutations in epigenetic regulators have a key role in the initiation and maintenance of hematopoietic malignancies and malignancies writ large. These mutations cooperate with other disease alleles to cause transformation. We now can use single-cell profiling to understand that process in humans, and we can go back to the lab to model that clonal evolution. And I think most importantly, we can go back to the lab and understand what these events do alone or in absentia and begin to do perturbations. And I'm most excited about the idea that all of this work is designed to identify therapeutic targets, and we're going to go back to the clinic, all working together in trials like the Beat AML trial that I've helped lead with the LLS and Amy Bird and Brian Drucker and John Bird and others. And we believe that that'll be important, and ultimately working with our colleagues, including at Dana Farber, to do prevention studies to prevent malignancies. So with that, I'll stop, and I'm happy to take questions. Nellie, I'll turn it over back to you. Thanks for allowing me to participate. And again, thanks to everybody in the lab and our collaborators for doing all of that work. Thanks so much.
Thanks, Ross, for the great talk. And we actually have a long list of questions. Looks like we have time. So let me start with the first one from Daniel Tennant. Is like, why did you leave out the most important mutation in AML, which is the CEBP-A? And then also the, why was it left out?
So it's a great question. Other than leaving it out just to spite Dan and get him to ask this question, in all seriousness, honestly, we focus on mutations in normal karyotype leukemia that had epigenetic regulators followed by other mutations. There are panels of CBP alpha. We think that's something that we need to do. We also need to figure out how to do copy number alteration. So we've largely obviated P53 mutant leukemia, because until we can do copy number in single cells, we can't map evolution. So lots to do there, but we had to start somewhere. And Dan, I promise we'll do that, hopefully with you.
Okay, so there are also a number of questions related to what is the effect of these mutations on stem cells? Like, does the order of event matter? Do they lead to expansion of particular cells? And my personal question also that, have you considered that these mutations basically just increase heterogeneity of the population without any particular target?
Those are all great questions. We know that phenotypically, that a lot of these mutations increase the number of and replating capacity, and in some cases, competitive advantage of primitive cells, whether it's stem cells or early progenitors. But the mechanisms by which they do that are largely elusive to date, and that's why we think we need new systems. Your point, Nellie, I totally agree. And so we think one of the intriguing things in some of these alleles, if you do the experiments many times, sometimes they win and sometimes they don't. And so I agree with you, Nellie, that understanding what is driving that heterogeneity, how important that heterogeneity is to the phenotype, and whether that can be perturbed is something that we as a field and we as a lab need to address moving forward.
Okay, and there's some questions also like, what genes do you want to target? These early genes or the progression genes?
I don't know.
Yeah.
I don't know. I think that's part of why we're doing all these functional systems that I talked about in the last third of my talk, is that I think there are arguments both ways. You know, IDH is an early event, whether it's an initiator in everyone or not, and it's definitely a validated therapeutic target, although there's clearly more mileage to get. So the idea that we shouldn't target the early events, I'm not sure. And then you could argue on the other hand that FLT3 is a really late event and maybe we shouldn't target it, but we just had gilteritinib approved. So I think we need to bring some sophistication to the approach and ask two things. One is, can we use things like clonal architecture to layer on top of mutations that maybe it's not just what gene to target, but where it is in the hierarchy in a patient? And the second is we need cocktails of medicines. I know that's obvious to this crowd, just like for Hep C and HIV, we're gonna need to ultimately target more than one mutation to get to curative strategies. So there's lots to figure out there, and I certainly have more questions than answers about that myself.
Okay, Bob Weinberg is asking you, like, have you considered the possibility of non-coding RNAs in addition to just the coding mutations?
Yeah, it's a great question. I guess our feeling was that we still have so much to do with the small number of coding mutations and combinations to keep our lab and other labs busy for the next few years, that we think that that is an important area that we're hoping lots of people are working on. We think, you know, I'll just give a shout out to my colleague Mike Karras or to Omar Abdel Wahab, who work both on RNA binding proteins and splicing. We think all of those mechanisms are relevant, but we got to start somewhere. And so we think understanding, we're going to focus on these mutations till we have a better sense of what they do. But we're hoping that people will take these models that we're developing and data we're developing and marry it with all those other data sets and approach it. And there'll be lots to do together.
Okay, other questions related to whether the, I mean, I think we already asked it, is the particular mutations lead to expansion of particular lineages, like for example, when the TET2 mutation is first and then NMP1 first is different, like they have a myeloid numbers increased. Yeah. Do you see a differences in the myelopoiesis?
Yeah, we do see some differences, and I think there's a lot we don't understand about that. I think one important question is, you know, to sort of take that to the next level is, how much do these mutations change the decisions that cells make versus change in early cells, the cells that are primed for different fates? Like, does TET2 instruct a cell, or does TET2 have a selective advantage in a cell that's already got a primed basis? And I think those are sorts of the questions that we need as a field to address, because although they're nuanced and detailed, they're really critical. Meaning, are you changing the blueprint, or are you actually transforming a cell that already has a predetermined blueprint, or some combination thereof?
So somewhat related to that, Jonathan Licht is asking you if there is actually such a thing as leukemia stem cell in AML, if you have a collection of clones, and if there is, like, what is that?
Well, I think, you know, I guess I've always been a little bit of a nihilist about leukemia stem cells. I think it's important not to be dogmatic on them. And we went into a lot of our early work thinking that it could be that a lot of it is due to things like the NOD-SCID assay and the xenotransplant barrier. But the reality is that in our work and a number of other labs, we've shown in, for example, the FLT3 TET2 model, that there is a hierarchy of the ability of the cells to propagate the disease and the ability to propagate other phenotypes. And I think the data with genotype and immunophenotype, that combination is going to really allow us to address it. And we need to get away from cells that do and don't have. For example, Martin Carroll had an awesome paper in JCI, you know, 8 or 9 years ago, showing that there's really a continuum of stem cell repopulating capacity. And so there may be variable amounts of stem cell identity in different clones. And we're hoping that some of these technologies will let us come back to some of those questions. That's a really good point.
Okay, another question related to the mutations, the pioneer mutations, you know, these early ones. Are those mutations actually could somehow predispose to the later mutation? Because, you know, if you have a mutation in the NMT, could you actually increase the frequency of mutations you can acquire in other?
So I think there's a few, yes, that absolutely could be true. And we had a paper in 2016 when Olga Gurianova was in the lab, who now has her own lab at University of Florida, showing that DNMT3A mutations can affect nucleosome remodeling and DNA repair. And there was also a paper, I think, from a group in Majewski and Peter Volk looking at DNA repair and DNMT3A mutations. So they can induce genomic instability. But I think there's a lot of different aspects to that. Do they induce instability? Do they enhance the fitness of specific combinations that they get selected out? Is there a bottleneck presented by the first mutation? You know, one thing that I didn't spend a lot of time in my talk, but if you look at our preprint, is that many of these later events are occurring over and over again, more than once, and that's not new. You know, Jerry Radich had a paper in PNAS a number of years ago with single cell mutational profiling with Martin Carroll showing that NPM1 occurs more than once in most patients. And I think that many of these mutations are being sampled over and over again to surmount specific bottlenecks that are, you know, presented either from cell autonomous, mutational, or non-autonomous mechanisms. And I think that that's an area that we in the field need to figure out was, you know, is transformation largely a result of trying to solve bottlenecks? Because why would you get 4 different mutations in one gene when, you know, and why is the rest of the genome not altered? And that has to be occurring for a very specific reason.
Okay, wow, we keep getting lots of questions. So question was, how important is the sequential acquisition of these mutations? What would happen if you have these DNMT, IDH, FLT3 mutation, and then you knock out one of the primary mutations? Would you still maintain the leukemia?
Well, that's a, you know, I hope whoever asked that question reads our next grant. Like, we think that that's the kind of question that the field needs to ask, which is, you know, ultimately, do these different mutations, does the order affect not only the phenotype, but the dependency? And for example, if FLT3 is occurring at different time points, does that affect the FLT3 dependency? That can be tested pharmacologically. And then I think some of these CRISPR approaches will let us, you know, go after some of the mutations where we don't yet have drugs. And so we totally, that's, you know, that's all we talk about in our lab, and we're excited during this break and moving forward to talk about it with the community more and more.
Okay, so a couple of questions related to how important the non-cell autonomous effects are, particularly the bone marrow niche, both in terms of, like, you know, AML is a disease of aging, and then also do the pioneering mutations actually can have non-cell autonomous effects that change the niche and then enable the clonal expansion and outgrowth?
Yeah, we think the niche is critical. I'll point out the really seminal work by Manu Pasage, who showed a number of years ago that CML, BCR-ABL positive clones can actually remodel the niche and render it inhospitable for wild type cells, similar, Nelly, to your own work in breast cancer, where one clone can affect another. We don't have similar work yet either with AML on precursor cells or on different clones, but we think obviously that's the functional direction the field needs to go. And I'll point out that we're reading all those papers, including some of your work, Nelly, because we think that there's a lot of lessons to be learned from that, to ask if the rules are the same in different contexts.
Yes, I feel like every mutation has non-cell autonomous effect, because especially epigenetic state of the cell changes that can change a lot of things. I mean,
yeah, I mean, you and I have both worked with Ron Fan and colleagues at Yale, right? And we think the ability to enumerate what individual cells or clones secrete is a really cool way to do it. And so we think that's another thing we can layer on, that we've done that both in mouse and human. And so the idea that you can do single cell secretion profiling, right, Nelly, is another powerful technology to think about.
Yes, actually, I think that was one of the questions here, that do you see any particular cytokine profile depending on a mutation?
Yeah. We haven't done a lot of cytokine profiling in leukemia. Some of you who know our work will know in myeloproliferative disorders, we've done a tremendous amount, but that's certainly one of the directions that we're beginning to go, and we were about to go before we had to go home like everybody else.
Okay. So question related to, do you think that this subtyping based on DNA sequencing is basically classifying patients into mutated, non-mutated, when you see that some of the mutations could be subclonal and you may miss it? So is this actually the best approach?
Ooh, well, I think, that's a lot of questions in one. Like, we don't know, for example, how clinically relevant is it to get down to these small clones? I don't know. I don't think we have any data yet in our lab to show that this has relevance at this point. We think it's an important question that needs to be addressed. I think likewise, we don't know biologically, what these small clones are doing and how important is it to know about it in each and every patient or each and every model, but we're sure as heck going to try to figure that out moving forward. So I think more known unknowns than known knowns on that one. Okay.
And a question related to DPM1 mutations, that the mutation seems to be, mutant wild type cells is very different phenotype, but then how do you see that this also predicts the clinical responsiveness to chemotherapy in the absence of other mutations?
Yeah, that's a really good point. The first thing I'd say is we don't know a lot about what NPM1 does. And there's been some really nice work from Peggy Goodell, recently Scott Armstrong's paper on really starting to give us some molecular determinants of what it's doing. But I think the point that question raises is that NPM1 has very different therapeutic and prognostic relevance with different subtypes. And that's, I think, a question that there's not really any sort of genomic or mechanistic data yet, and we really need to understand. So I agree with that question, and we certainly hope that some of the models we're developing, if they're chemo responsive versus resistant, and they recapitulate, for example, an NPM1 TET2 model, if it's chemo responsive, and an NPM1 FLT3 it's not, we'll hopefully be able to shed some light on that, but lots to do.
Okay. Golam Mohi is asking you how many mutations you need for AML, because it looks like you have 3 mutations, but then sometimes 2 mutations is already enough.
I think it's really hard to say because, ultimately we think that in human AML, it's 3 to 8 coding, with the caveat of Bob's question that non-coding and RNA and all those things we think are also important. We think it's probably in that ballpark. The problem with mouse models, I'll remind all of you, is a couple of fold. The first is that when we engineer a mouse model, in general, we're putting the first mutation in every cell, and usually the second and third in most cells, unless you're doing a really careful sort of like very limited, Cree exposure. And so what you're doing is seeding a mouse with thousands, if not tens or hundreds of thousands of clones that have these mutations. So it's the wrong question to ask in the mouse, because if you say, well, 3 mutations gives me AML in every mouse, it could be that it's because if you put 3 mutations and 1,000 clones, that gives you leukemia in every mouse. That may not be the same in humans. You may need more mutations. I'll point out work from Ben Ebert's lab where they've done CRISPR'd lots of mutations, both in mouse and human, and shown it's actually a lot less efficient, suggesting potentially you need more events to occur, and that it's a little bit more stochastic than these deterministic models suggest. So I think there's a lot to do there, and I think that hopefully people that really model this can help us figure that out. So I think I don't know the answer to that. It's something we talk a lot about. And I think it's best talked about on Friday afternoon over a beer, or maybe this week for me over some kosher Passover wine.
Yeah, so somewhat related to this is, have you done single cell RNA-seq studies in the mouse models to see how well they reproduce the heterogeneity?
Not a lot, Nellie. I think we're just starting to do that. We were a little late to our single cell RNA sequencing. Part of it, honestly, because a lot of our earlier models were, again, putting all the mutations in together, Nellie, and we thought those weren't the models to spend a tremendous amount of resource doing single cell profiling. The models we have now, if you have Cree and FLT, we not only have those in different orders, just like humans, they have reporters. So we can actually read out for every cell, not only what its transcripts are, but what mutations it has. And so we're now doing that, and we thought it was the right time, whereas a year ago, although we had earlier models, it wasn't the right time.
Okay. The other thing is, like, I don't know if you know the Jim Gregory's, very nice experiments to look at the age effect. Like you would put in this.
He's got awesome. He's got awesome data. I will tell you that a lot of my thinking on a lot of aging and cancer was impacted, as Jim knows, from when I went out there and gave talks 6 or 7 years ago, and he was talking about evolution. And I agree. I think his models and the work he's done is really awesome. And we've got a little bit of work, mostly at looking at some of the early mutations in aging with a grant from the Waxman Foundation. But I think there's just so much to do to layer aging in. And I've been a big proponent that we need, as we come back from this hiatus, to think of ways as a field to layer in more aging work in our models, because it's just really resource intensive to age all of these models, and we need to do this collaboratively, because if everyone ages all their mice independently, it's going to be a not ideal use of resources.
Yeah. Okay. So a couple of questions related to how would you consider, like, for treatment, the subclonal diversity? Does it impact the likelihood of response? And then would you target the major clones or at once or sequentially to get the best effect? And then lastly, is the number of mutations and the clonal diversity influence the immunity against the disease?
So those are all great questions that we cannot yet address that we think need to be addressed. Certainly, we're very interested in asking, does therapeutic response in humans and maybe in the models affected by clonal diversity and targeting different clones? We're going to need large access to large, homogeneously treated cohorts to do that. And hopefully, we've got those samples out there in the world and, the cooperative groups in other settings, and others will do similar work. The questions about immunity, it's a great one. I'd love to talk to folks who have ideas on that and find ways to collaborate. We definitely are most interested for our own purpose in the cell surface repertoire more than immune visibility, which I think is an area that lots of other folks are working on. And so we're very eager to collaborate with people and brainstorm on that.
Okay. And there are some couple of technical questions. Is, for example, when you're doing single cell analysis, should you enrich for CD34 positive cells to get a higher depths for the progenitors?
Yeah, so all we did was do a BT depletion. We didn't do 34 because a lot of these are NPM1 mutant, and the leukemia aficionados will know that NPM1 mutant are 34 negative. So we think that you'll want to do studies like that. But our hope is as the cost goes down and you'll be able to get more cells, hopefully as you marry the antibodies in and you marry the genotype with the immunophenotype, will obviate some of that. But I do think you're going to want to do some sorting and sequencing because it'll maximize the amount of information you get for very specific questions.
Yeah, somewhat related to that is why has AML hasn't been responsive to immune checkpoint therapy?
Well, it's pretty low mutational burden, right? These patients are pretty old and pretty sick. I think there's a lot of reasons why that is. And obviously Matt Davids and Rob Seufer from your institution have shown that, CTLA-4 blockade post-transplant showed efficacy, suggesting potentially if you give it an antigen like an allo, we can engender immunity, and transplant's been around forever. So I believe it can be immunologically visible, but the question is whether the disease without that can be visible needs to be addressed, and the jury is still out.
So somewhat related to that, do you think that all these mutations that we're detecting in normal tissues, people without disease, do you think that's just basically to kind of hijack the immune system, and then by the time the tumor starts, you actually don't have an immune response?
I think it could be a factor. I've always thought that, at least in the hematopoietic compartment, that the primary factor is that these cells appear to be, enhanced in their fitness. And when you have stressors in life, infection, inflammation, immune challenge, that these mutant cells are just more resilient and they're able to survive each of those stressors. But I don't think it's purely immune visibility, but that's just an opinion, Nelly.
Okay. I think, there are still a number of questions that I have that you can look at it later on.
Yeah, people can tweet them to me, and I'm happy to chat. And I hope people have lots of ideas.
Yeah, lots of ideas, lots of suggestions, which is great. Okay, so with that, again, I'd like to thank Ross for the excellent talk. And as a reminder, the website, the DFHCC website will have the slide presentation uploaded, so you can look at the presentation later on. And again, he will answer the questions. The Q&A will be available to him as well. And as again, the reminder also the change in the Zoom link that Richard announced at the beginning, and that will also be posted on the website. So from Monday, we have we're using a new Zoom link to the seminars. So thanks again for everyone for participating, and Ross for a great talk, and have a great weekend.
Thanks for including me. Thanks so much.
Thank you. Bye-bye.